Transport through membranes. This topic is so extremely important. This one is such a massive topic and it comes up every exam. I can't guarantee that for sure, but it comes up almost every single year in GCSE and you need to know it well. So let's break it down. There's diffusion, osmosis, and active transport. Diffusion is, you need to know all the definitions by heart. So diffusion, defining it, is net movement of particles in solution or a gas form from an area of a high concentration to an area of a lower concentration. So from high to low, essentially. There are factors that affect diffusion rate that you also do need to know because sometimes they ask you to apply it to the question. So here we talk about steepness of concentration gradient. So imagine you have, let's say, a bunch of particles going around and there's a membrane. There is, let's say, 80% of particles on the right and like 20% of particles on the left. That's going to move very quickly and have a very fast diffusion rate because there's such a steep concentration gradient. So if there were only 30% of particles on the right and 20 on the left, it's not going to be as quick because it's not as steep. So the steeper the concentration gradient, the faster the diffusion rate. Temperature works similarly. The higher the temperature, the faster the diffusion rate, higher the diffusion rate, because higher temperature, more energy. Surface area, similar again. The larger the surface area, the higher the diffusion rate. Diffusion distance is the opposite. The shorter the diffusion distance, the higher the diffusion rate. And the reasoning, look at the diagram in the bottom left corner, you can see that there are the capillaries. And it is the alveoli, and it's just doing gas exchange. The capillaries are 1 cell thick. They are very thin. And because think about if you're moving stuff from very thin, diffusing stuff, like moving stuff over, it's going to be a lot easier than transporting very thick layer. Think of it like a very thin towel. If you have a very thin towel and you pour water over it, more water is going to come through if it's thinner. If it's very thick, there's not going to be as much water going through. So that's kind of how you would think about when it comes to diffusion distance. So the shorter the diffusion distance, the higher the diffusion rate. One thing I want to point out for those who are doing AQA, you do actually need to know more specifics on diffusion. So things like intestinal villi or alveoli, you need to know in detail on what the adaptations are. So I'll just quickly go through that with you. If it's alveoli, capillaries, or whatever, they are 1 cell thick. They're very thin, so short diffusion distance. Alveoli do have a very large surface area. Same with villi, they have a very large surface area. And they also, for villi, they have a lot of microvilli, increase the surface area. That's just kind of what you need to know for AQA specifically, but generally everyone else does need to know the factors affecting diffusion rate. This goes with everyone else. Right. Let's move on to osmosis. This is where people get a little confused because they're very similar. Osmosis is a type of diffusion, and osmosis is movement of water. So if you think about it, the osmosis is diffusion of water from a high water concentration to a low water concentration. A high water concentration meaning dilute solution, and a low water concentration meaning a concentrated solution. So if you were to write the definition of osmosis, it would be the diffusion of water from a dilute solution to a concentrated solution. And that's one part of it. Second mark, people always forget, you have to write this down, is through a partially permeable membrane. When you're talking about osmosis, you have to talk about through a partially permeable membrane. Okay, there's an experiment that you guys might have done for osmosis, but I'm not going to go through the details of the experiment, but I will talk about like the application of it. So osmosis, usually cells can be put in solutions with different relative concentrations. And there are 3 things you need to know with those concentrations and the relative concentrations. First one is isotonic, where the cell is put into a solution that has the same concentration as the cell. Isotonic. There's no movement because the concentration is the same inside the cell and outside the cell. This is not written on here, but just to tell you the term, just because sometimes they do use that term isotonic. Same with hypertonic, hypotonic, they will give you these terms. Hypertonic, things like a salty brine. So a very concentrated solution. So it's more concentrated outside the cell than inside the cell. Hypotonic is the opposite. it is when it is very, very dilute, things like pure water. So it's going to be more concentrated inside the cell than outside the cell. Okay, I'm going to walk through why you need to know this. In a hypertonic solution, where it's very, very concentrated in the very, very concentrated solution, and the cell is not as concentrated as the solution, because there's so much water inside the cell and it's less concentrated, so more water in the cell, water is going to want to move out of the cell through osmosis. Through this, because there's water moving out, cells shrink. In animal cells, we use the word shrunken or shriveled. For plant cells, we use the word plasmalized. So it starts flaccid and it becomes plasmalized. It shrunk. Hypotonic is when it is the opposite, because of things like pure water. It's going to be more dilute on the outside. So water is going to move from the solution into the cell through osmosis because there's more water outside. This is going to cause the animal cells to swell up. And sometimes, because there's so much water going in, it's going to burst. In plant cells, we use the term turgid for describing cells that are filled up with a lot of water, essentially. However, it's good to think about why plant cells don't burst, because plant cells have a cell wall. They have mechanical strength, therefore they do not burst compared to animal cells that will easily burst because there's so much water going in. They don't have a cell wall. So that's something to think about. But you do need to know the three different types. Isotonic, same concentration. Hypertonic, solution is more concentrated. Hypotonic, solution is more dilute. Okay, let's move on to active transport. With active transport, the definition is basically the exact opposite of diffusion. It's the movement from a dilute solution to a more concentrated solution. So not dilute solution of water. So it's a movement from a less concentrated to a more concentrated solution. So it's from low to high. Diffusion is high to low. Active transport is low to high. And this is the first mark when you're defining active transport. Second mark, you have to talk about how it requires energy. In the name itself, active transport, it is active. It's an active process. Therefore, you need energy for any movement. And this energy comes from respiration. So the examples they use a lot, and I would say that you probably do need to know, is two things. Absorption of sugar molecules. So usually sugar goes from a low concentration in the small intestines to a higher concentration in the blood. Second one will be the absorption of mineral ions. Usually there's a very, very little mineral ions in the soil. And because plants need those mineral ions, they're going to go from a low concentration in the soil up into the plant root hairs. Plant root hairs, they have a large surface area that allow them to absorb these mineral ions very, very easily. So that's just one adaptation. But essentially, it goes from a low to high concentration into the plant root hairs. Okay. That is transport through membranes. We're going to quickly look at a question here. This applies more to AQA, if anything. As I've said, AQA does ask you to look at the alveoli and villi, and that you need to know the adaptations. So I'll just work through this really quickly with you. This is AQA, you need to memorize it, but everyone else who does all the other exam boards, you still need to know that if they tell you there's a large surface area here, you need to be able to figure out, oh, the higher diffusion rate, more absorption. So in this question, it's asking you how the alveoli and the villi are adapted to increase absorption. The question itself, you just need to talk about how they have a large surface area, which allows it to maximize diffusion. So villi and alveoli both have very large surface areas. Especially with villi, they have these microvilli. They also have very short diffusion distance, because they have very, very thin walls. So again, thin walls, short diffusion distance, higher diffusion rate, better absorption. Something that I feel like is also good to note that they don't mention within diffusion that applies specifically to alveoli and villi, so our biological structures. They have very good blood supply, or I would like to say capillary network. So they have a very good capillary network allowing them to maintain a very good concentration gradient. An extra bit of information as well that is actually good for application in biology is that villi, they have a lot of mitochondria. Because they have a lot of mitochondria, that allows them to have a lot of energy. So for active transport. So that's transport through membranes. Just, it's important to note all of that, essentially. I don't think there's any more that you need to know within transport through membranes when it comes to content. It's more about knowing how to apply the apply your knowledge to the questions. These are very application heavy, so know your basics.